Thank you for the honor of being able to make this presentation on gene silencing strategies for HIV control. And I'm going to concentrate on the use of si and sh RNAs. These are my potential conflicts of interest. I'm an inventor on a number of patents associated with this approach, and we get some help from both Calimmune and CSL for supporting the prosecution of those patents. As we all know, HIV drugs have revolutionized HIV treatment, but they only hold the virus in check. They are not a cure in themselves. They reduce plasma viremia and viral load to levels below detection. And it reduces the amount of viral DNA in CD4 T cells and in myeloid cells, but it does not clear the DNA from those cells. The integrated viral DNA persists as a reservoir, and this reservoir can persist for decades despite effective antiretroviral therapy that suppresses the plasma viral load to levels below the level of detection. In the reservoir, the DNA form of the virus is mostly in a latent state. That is, it's not transcribing. However, if you stop antiretroviral therapy, the plasma viremia returns from the reservoir, usually within weeks or at most months of stopping the antiretroviral therapy. Therefore, any approach to cure needs to consider the control of the reservoir, and this will be an essential component of any cure or remission strategy. Before we go any further, I think we need to understand a little bit more about what the latent state of the virus is. The most important latent state in terms of the reservoir is this form of post-integration latency. That means that the HIV is in its DNA form, it's integrated into the host genome, but it's not transcribing. That is, it is not making RNA. And in all host genes, transcription is driven by promoters. And so every gene in the human genome has a promoter that drives ' transcription. And in HIV, this promoter is known as the 5' long-term 5' LTR. And there are several mechanisms of latency. The most important form in terms of this presentation is epigenetic latency. That is, this is regulated mostly by the histone architecture, and particularly the histone architecture within the 5' LTR, or long terminal repeat. And this histone architecture depends in turn on the chemistry of the histones and whether those histones are either acetylated or methylated. And I'll come back to that in a moment. Epigenetic silencing also depends on whether there's the presence or absence of host transcription factors and or transcription factors that are produced by the virus, such as TAP. And if they're sequestrated, then that prevents the upregulation of viral transcription. There are other mechanisms by which HIV might be latent. It might have integrated into a silence gene and so be in the context of closed chromatin, or in fact, it may be in the form of a defective virus that is not replication competent, that may have a defect in its 5' LTR and therefore unable to transcribe. This is a cartoon of the HIV genome showing the complete genome above in various colors here, and then blown up representation of the 5' LTR. There are a couple of very important things to note. The 5' LTR has a series of binding sites for a range of host transcription factors, and these are shown in various colors here. One to note, for instance, is the NF-kappa B binding region, which is an important host transcription factor that upregulates HIV transcription. And then there are multiple other host binding sites for other host transcription factors such as NFAT and AP1. The host transcription factors bind to the 5' LTR and upregulate HIV transcription. The start site is shown here. The HIV DNA is associated with nucleosomes in the same way that human DNA is associated with nucleosomes. And in the 5' LTR, there are 2 nucleosomes shown here, nuc0 and nuc1. The nucleosomes are made up of various sorts of histones, and the DNA winds itself around these nucleosomes. Changes in the chemistry of these histones, that is, whether they're acetylated or methylated, determines whether this region of HIV DNA is actually compacted into compacted chromatin or open and accessible. If it's compacted, it is difficult for the host transcription factors to bind to upregulate HIV transcription. If the chromatin is open and accessible, and that's associated with histone acetylation, then the host transcription factors more easily bind. So there are a number of ways that have been attempted to undertake cure strategies. And these can be summarized into 2 forms. One is called shock and kill, and the other I'm going to refer to as block and lock. Both of these manipulate the epigenetics of the 5' LTR. And essentially they change the acetylation and methylation profiles of the 5' LTR of the virus and either open up or close the chromatin. And in shock and kill, the idea is that various small molecule inhibitors such as histone deacetylase inhibitors or histone methyltransferase inhibitors open up the chromatin and allow HIV to transcribe. The HIV proteins are expressed on the surface of the cell. If antiretroviral therapy is present, then the virus can't be produced, and the presence of HIV proteins announces these cells to the immune system and they're killed. And so that means that the reservoir should be reduced. And this is most of the trials that have been done in the cure sphere have been in the sphere of shock and kill and have had limited success so far, both with waking up the virus and with killing the virus when it is woken up. The alternative approach, which is what's going to be the subject of the remainder of this lecture, is this block and lock approach, where we do exactly the opposite. The virus is already in a latent state, and we try and lock it down into a latent state by reinforcing the methylation of the histones and stopping the acetylation of the histones. And the way we've attempted to do this is using si or shRNAs, but there are other ways of doing this with small molecule inhibitors and also with TAT mimics. The idea is that this shuts down HIV transcription into a form and induces a form of super latency, which the virus cannot wake up from. So I refer to this as either bind and gag or block and lock. And the idea is that you take the open form of the HIV chromatin and you use an siRNA that binds to the 5' LTR. It accesses this transcriptional silencing complex called the RITS, and it induces this state of closed chromatin in the 5' LTR and forces the virus into a latent state. If the virus is already in a latent state, it maintains the virus in its latent state. So we have identified a number of si or sh, that is short hairpin RNAs, that target various sequences in the 5' LTR. 2 are shown here in this cartoon. One is called siPromA, which was the first one we described, which binds to a region that overlaps with the NF-kappa B binding region. And a second one that I'm going to describe in this lecture is called si143, and it binds close to the AP1 binding site within the chromosome. One of these is not associated with the nucleosome binding site, and one of these is associated with the nuke 0 binding site. Both of these sequences that are encoded by the siRNA are unique to HIV. They do not have similar sequences in the human genome, and importantly, they're relatively conserved within the HIV genome. And in fact, we've described many more of these now, up to 14 or 15 of these potential sequences. And we know that if we use 4 of these sequences, we can get coverage of over 99% of the variants of HIV that are described in the Los Alamos database. So we can both get specificity of this approach through nucleotide binding, but we can also get coverage of all the viral variants using this approach. This is an in vitro experiment showing viral replication as measured by reverse transcriptase production and the days post-infection in a cell line that's replicating every day, reproducing the virus. These are the controls here shown in the black lines, and you can see that the virus replicates very nicely in this culture. But with either the siPromA in red or the Si143, the sequences of which are shown here, you get profound silencing of the virus. If we were measuring RNA, we can show that we get a 3 to 4 log reduction in the amount of virus that's being produced, and that the suppression is prolonged, lasting up to 21 days after a single exposure to an siRNA. So this causes prolonged epigenetic changes within these cell lines that force the virus into prolonged levels of latency, even though the siRNAs are probably only surviving in the culture for a day or two. The effects of those siRNAs last for a much longer time. We've shown that we can induce this effect in multiple T cell lines, in monocyte-derived macrophages, and importantly, in astrocytes, as well as in primary CD4 T cells. And we have shown that these effects are highly specific with minimal or no off-target effects. Furthermore, if these molecules are in a culture and the culture is silenced, the virus has great difficulty waking up. And even if we try and wake the virus up with something like vorinostat, which is one of those HDAC inhibitors that is used in a lot of shock and kill trials, and the concentration of that is shown here. Or if we use TNF, which is a strong T cell activator, we get the presence of the siRNAs cause prolonged silencing of the virus, even the presence of these molecules that are waking up the virus, and even at concentrations much higher than those that are physiologically present in septic shock, TNF, or in the pharmacological concentrations used in clinical trials shown here. So we can induce something like super latency. Furthermore, this works in vivo. This is, we've used humanized mouse models of acute HIV infection. And with the presence of the PromA in CD4 cells or in stem cells, we get decreased viral production and maintenance of CD4 T cells. We have taken this further and used a combined approach, mimicking combined antiretroviral therapy, where we combine a siRNA that knocks down the chemokine receptor CCR5 and combined with our PromA. And this gives us dual blocking. It stops the virus getting into the cell, and it also stops the virus reproducing and closes down the chromatin. This is a schema of another mouse model experiment where we've done where we treat the mice with antiretroviral therapy and then interrupt in an analytical treatment interruption. And the color code is that the blue are the controls, the red is the PromA alone, and the green is the combined anti-CCR5 and PromA gene therapy. And you can see here that over time, that in the combined therapy, there is maintenance of the CD4 T cells even after the withdrawal of antiretroviral therapy, whereas in the controls, there is a distinct decrease in CD4 T cells. Furthermore, there is a delay in both the treatment groups of viral rebound after antiretroviral therapy relative to the controls. So we can inhibit recrudescence of the virus from the reservoir using these siRNAs, not just in tissue culture, but in vivo, in these humanized mouse models. But the effect is relatively limited, and it's relatively limited because we get relatively low transduction of our stem cells or our CD4 T cells. And we need better ways of delivering the gene payload. And so we've looked at this by making new retroviral delivery constructs using particular envelopes of HIV that allows us to get much better delivery into a range of CD4 T cells. And we've also increased the ability of these siRNAs to be transcribed by, again, using VPX, which allows better transduction and transcription of these siRNAs from both resting CD4 T cells and macrophages. And our primary candidate is shown here, where we get a 10-fold induction of increase in transcription of our siRNAs using this particular VPX. We are also using a range of nanoparticles to deliver this payload because this may allow us to give the siRNAs directly intravenously. This is how we make the nanoparticles. The siRNAs are incorporated into these layer by layer nanoparticles, which we are making with the University of Melbourne. And even without targeting of these these nanoparticles to CD4 T cells, we can get transduction of 15% of arresting CD4 T cells, which is a very difficult target for gene therapy. With standard gene therapy, you will get 1 to 2% of resting CD4 T cells transduced with a VSV-G pseudotyped retrovirus. And so even without targeting, we're getting 15% of resting primary CD4 T cells transduced. And we believe this is a promising delivery system for our siRNAs. So in conclusion, I think I've demonstrated that shRNAs that target controlled regions of the HIV promoter induce prolonged and profound and specific silencing. We can do this in multiple cell types where the reservoir is found in vivo, including T cells, macrophages, and astrocytes. And this, the latency that's induced is highly resistant to reactivation. These in vitro findings can be replicated in vivo in 2 separate murine models, but we need to do better at delivery of the gene therapy. And we're looking to build better retroviral delivery vectors by modifying the vector, not activating the cells, and also using a range of novel nanoparticles to deliver these gene therapies. This work has been conducted in collaboration between the Kirby Institute, led by Chantal Allensteil. We've worked closely with the nanoparticle engineers at Melbourne University, including Frank Caruso. The mouse work has been done both at Kunamatu University and at the Walter and Eliza Hall. And we work closely with CSL and Calamine in terms of the gene therapy delivery constructs. Thank you.